Oh, hello everyone. It's been such a what a day. Oh wow. Wait, guys. Oh, okay, wait. Okay. So let's see. Can you guys hear me? Hello, hello. Hello. Hello, Salati. Hello, people. Fine people. Okay, so today we're going to be going over. Hold on. Let me bring up my AP bio. Gonna put in tiles here. Window. Move resize. Top right. Right here. Perfect. Okay, I'm actually gonna extend this over here so you guys can see that better. Make that really small. There. Alright. We're gonna go over I'm struggling to open this right now. Protein folding assignment, okay? We're gonna be learning about protein folding. Empty my trash real quick. We're gonna read it off of this. So, an arrow on two different peptide bonds. If you guys know what peptide bonds are, they are there to link different amino acids together. Or amino acid chains are linked up together by peptide bonds, which make polypeptides. And protein structure and function. Okay, the specific activities of protein result from their intricate three-dimensional architecture. Okay. The simplest level of which is the sequence of their amino acids. What can the amino acid sequence of polypeptide tell us about the three-dimensional structure, commonly referred to as simply the structure of the protein and its function? The term polypeptide is not synonymous with the term protein, even for a protein consisting of a single polypeptide. The relationship is somewhat analogous. I don't know what that says. Let's actually look it up right now. Look up. Colors are groups of colors. Colors. What? We're just gonna ignore that. We'll start studying for the SAT in about 30-20 minutes. Okay. But one or more polypeptides precisely twisted, folded, and coiled into a molecule of unique shape, which can be shown in different ways of models. Figure 3.20. Or I think I'm actually a little bit far off. Here, this is where I was last time. Here, N-terminus, which is one end of the polypeptide chain, and a C-terminus, which is the other end. And it has a free carboxyl group. Cool. Nope, don't want to know about that. The free carboxyl group right here. Highlight. The C-terminus. The chemical nature of the molecule as a whole is determined by the kind and sequence of the side chains, which determine how a polypeptide folds and thus its final shape and chemical characteristics. The immense variety of polypeptides in nature illustrates an important concept introduced earlier, that cells can make many different polymers by linking a limited set of monomers into diverse sequences, making a polypeptide chain. Okay, so right here we can see that's dehydration synthesis. It takes out water, which is dehydrating it, and then a peptide bond is formed. Peptide bonds are formed by dehydration reactions. Here we go. Right there. Which can link the carboxyl group of one amino acid to the amino group of the next. So this would be an amino group right here. Two different peptide bonds. So peptide bond would be between the carbon and the nitrogen. Let's see. Peptide bond between. Let's see if we can find where the peptide bonds are. You guys can see this, right? The updates. Right here. You guys see that? Is it updating? Please update. No, it's not updating. I need this to sync. Please sync this notebook. Okay, sorry about that, guys. A little bit of background noise. Let's see. Okay, I was actually in the wrong one here. Just switched over. So it tells us to draw a circle around two of the amino acids shown in the picture below. Draw an arrow pointing to one peptide bond. I'm assuming it's going to be carbon and the nitrogen. Oh, here's a carboxyl group. I can see a carboxyl group right there. So one carboxyl group is connected by carboxyl groups, right? Carboxyl groups, I forgot. Carbon and a double bonded to an oxygen atom. Okay, so it's oxygen double bonded to carbon with an OH. Okay. With an OH. Let's see here. Let's bring in my notes too. For the SAT prep. Let's see. I think the bond is right around here. Here is our carbon double bonded to oxygen and our nitrogen here. Or it should be an OH right here, actually. Or OH is probably somewhere. Just make sure you guys can see that. Two of the amino acids shown in the picture below. Arrow pointed to one peptide bond. Okay, so the OH bonds to it seems to always bond to a nitrogen here. So I'm gonna assume, okay, so I think this is one right here. This is one peptide bond. And then this would be the long chain of carbons for I think it's the side chain. I think that's what it is. Don't take my word for it. Let's see. So we have another carboxyl group here with an H right here, bonded to to what exactly? Wait, yeah, there's another. I think there's another peptide bond right here. This might be another one. It's another carboxyl group. There's so many carboxyl groups actually. There's plenty. So all my carboxyl groups right there, I think. So around two of the amino acids pointing to one peptide bond. Okay, so there's just an arrow, just pointing to one. So I'll just draw an arrow right here for the peptide bond. This should be one peptide bond. Should be always next to the carboxyl group, I think. I'm gonna have to ask my teacher just to make sure. Let's see. Okay, so draw a circle around two of the amino acids shown in the picture below. Okay, so we must have an amine group and a carboxyl group. This is what it always must have. Okay, so here's our amine group. This is one amine group right here. And here is our carboxyl group. So I think this is one amino acid right here. This is one. It's connected to another one, and then this one is our second amino acid. I think those are two amino acids. What level of protein structure, primary, secondary, or tertiary, or quaternary, is the diagram above showing? Describe slash define this level structure. Okay, so we're not actually there yet. So we're gonna keep reading. The specific activities of proteins result from their intricate three-dimensional architecture, the simplest level of which is the sequence of their amino acids. Yeah? Excuse me, I have to take my doggo out to potty. Give me 1 second. Back in a couple minutes. Just gonna make sure. We'll be back in a couple minutes. You're back. Okay. Mom! You're back, guys. Okay, guys, I'm back. Okay. We're gonna continue our protein structure assignment. Don't worry, the cool SAT prep will come soon. I have to at least understand this stuff. Okay, so the simplest level, which is the sequence of their amino acids. What can the amino acid sequence of a polypeptide tell us about the three-dimensional structure, commonly referred to simply as the structure of the protein and its function? The term polypeptide is not synonymous with the term protein. Even for a protein consisting of a single polypeptide, the relationship is somewhat analogous to that between a long strand of yarn and a sweater of particular size and shape that can be knitted. What is synonymous? Let's look it up. the same name is not the same as protein. That's what they meant. Okay, functional protein is not just a polypeptide gene, but one or more polypeptides precisely twisted. Functional protein is not just a polypeptide chain, but one or more polypeptides precisely twisted, folded, and coiled into a molecule of unique shape. That's our definition here, which can be shown in several different types of models. Figure 3.20. And it is the amino acid sequence of each polypeptide that determines what three-dimensional structures the protein will have under normal cellular conditions. Okay, visualizing proteins. 3.20. Here we go. Excuse me for that background noise, guys. Okay, so AP apply concepts and connect across big ideas using visual representations. What determines the properties of a protein? What structural properties are critical for membrane-bound transport proteins? How will it change in a DNA protein, or how is a change in a protein and gene expression related to a change in DNA sequence? Let's take a look at this photo here. Okay. Okay, guys, I'm back. Sorry, I forgot to take my dog out. So there's our polypeptide chains. We're going to continue on with our protein structure study. Okay, using data from structural studies of proteins, computers can generate various types of models. They can generate various types of models. Each model emphasizes a different aspect of the protein structure. But no model can show what a protein actually look like. These three depict lysosome, lysozyme, a protein in tears and saliva that helps prevent infection by binding into target molecules on bacteria. In which model is it easiest to follow the polypeptide backbone? Okay, space-filling model emphasizes the overall globular shape, shows all the atoms of the protein except hydrogen. Hello, Sohail Kazi. Cool. Hello, viewer. We're gonna study for the SAT in 15 minutes. Ribbon model shows only the polypeptide backbone, emphasizing how it folds and coils to form a 3D shape, in this case, stabilized by disulfide bridges, which is the yellow lines. Okay. Target molecule wireframe model shows the polypeptide backbone with side chains. Hello, Bushra. Wait, why are all the never mind. I'm not gonna say it. Hello, everyone. It's nice to have viewers. Nice to have people watching. Okay, a ribbon model purple is superimposed on the wireframe model. The bacterial target molecule yellow is bound. Okay. Hella quiet. I probably have to put the microphone gain up a little bit. Hopefully it's a little bit better. Simplified diagrams. It isn't always necessary to use detailed computer models. Simplified diagrams are useful when the focus of the figure is on the function of the protein, not the structure. A transparent shape is drawn around the contours of a ribbon model of the protein rhodopsin, showing the shape of the molecule as well as some internal details. When structural details are not needed, a solid shape can be used. Simple shape is used here to represent a generic enzyme because the diagram focuses on the enzyme action in general. Okay, that makes sense. Pancreas cell secreting insulin. Okay, sometimes a protein is represented simply as a dot, as shown here for insulin. I see. Okay, so there's certain diagrams and models, not diagrams, there's certain models that are represented in different ways for different reasons, is what I'm getting out of this image here. Okay, when a cell synthesizes a polypeptide, the chain may fold spontaneously, assuming the functional structure for that protein. This folding is driven and reinforced by the formation of various bonds between parts of the chain, which in turn depends on the sequence of amino acids. Many proteins are roughly spherical, globular proteins, while others are shaped like long fibers, fibrous proteins. Even within these broad categories, countless variations exist. A protein's specific structure determines how it works. In almost every case, the function of a protein depends on its ability to recognize and bind to some other molecule. In an especially striking example of the marriage of form and function, figure 3.21 shows the exact match of a shape between an antibody, a protein in the body, and the particular foreign substance on a flu virus that the antibody binds to and marks for destruction. You can see it's very, very similar here. So what does this end right here? It's called exact match of shape between the antibody. It's just the shape, I guess. One of the fluid antibody binds to marks for destruction. Complementarity of shape between two protein surfaces. Protein from flu virus and antibody protein. A technique called X-ray crystallography was used to generate a computer model of an antibody protein, blue and orange left. Okay, so this is the antibody here, and the flu virus protein. This is a wireframe model modeled by adding an electron density map in the region where the two proteins meet. Computer software was then used to back the images away from each other slightly. Cool. See about the two proteins. Another example of molecules with matching shapes is morphine molecules, a manufactured drug, and endorphin molecules named for endogenous produced by the body, morphine. Both fit into the receptor molecules on the surface of brain cells in humans, producing euphoria and relieving pain. Morphine, heroin, and other opiate drugs are able to mimic endorphins because they all share a similar shape with endorphins and can thus fit into and bind to endorphin receptors. This fit is very specific, something like a handshake. See 2.14. Nope, this is a little bit too far. Scroll way down here. Okay, this fit is very specific, something like a handshake, the endorphin receptor like other receptors of protein. The function of a protein, for instance, the ability of a receptor protein to bind to a particular pain-relieving signaling molecule, is an emergent properties resulting from exquisite molecular order. 4 levels of protein structure. In spite of their great diversity, all proteins share 3 superimposed levels of structure, known as primary, secondary, tertiary structure, and tertiary structure. A 4th level, quaternary structure, arises when a protein consists of 2 or more polypeptide chains. Figure 3.22 describes these 4 levels of protein structure. Be sure to study this figure thoroughly before going to the next section. Okay, let's take a look here. Take a big look here. Primary structure, linear chain of amino acids. Let's see. Okay, here's our carboxyl group. Here's our amine group. Yes, I do have to have that SAT coming out next week. October 5th, I have to lock in, you know. I'm locking in this year. I cannot fall off like I did a while back. Okay, anyways, okay, so here's different amino acids all chained together. Primary structure of this amino end would be called like something, I forgot what it was called. Anyways, oh, here's your carboxyl end. So it always ends in a carboxyl group. What was it? O minus. Okay, the primary structure of a protein is a sequence of amino acids. As an example, let's consider transthyretin. Transthyretin? Yeah, so this one right here. A globular blood protein that sorry, I'm a bit nasally, guys. It's kind of hard for me to speak a little bit. I'm trying my best. Anyways, transthyretin is made up of 4 identical polypeptide chains, each composed of 127 amino acids. Shown here is one of these chains unraveled for a closer look at its primary structure. Each of the 127 positions. Good night. See you later, Gabo. Thank you for the good luck wish. Each of the 127 positions along the chain is occupied by one of the 20 amino acids, indicated here by its three-letter abbreviation. Yeah. Wait, three-letter abbreviation where? Oh yeah, here's Okay, that makes sense. There's 20 amino acids. It's oftenly used in, or no, only 20 amino acids is used in animal cells. No, not animal cells. Used in animals. Okay, the primary structure is like the order of letters in a very long word. Okay. If left to chance, there would be 20 to the 127 power different ways of making a polypeptide chain, 127 amino acids long. However, the precise primary structure of a protein is determined not by the random linking of amino acids, but by inherited genetic information. Oh, interesting. Okay, the primary structure in turn dictates secondary and tertiary structure. Due to the chemical nature of the backbone and the side chains are group of the amino acids along the polypeptide. Regions stabilized by hydrogen bonds. Secondary. A beta pleated sheet. Alpha, a region of an alpha helix in a transthyretin. Hydrogen bonds. Cool. Excuse the background noise again, guys. It's a little bit obnoxious, to be honest. Okay, beta strand, often shown as a folded or flat arrow pointing toward the carboxyl end. Plated sheet. So what's the difference between primary or linear chain? Secondary. Primary, secondary. What's the difference between primary and secondary? Let's, of course, look it up. I actually don't need this anymore. Ooh, particle accelerator. Hold on, guys. I gotta check out this Minecraft particle accelerator by FunkyJ13.
That I shut off in the lab and further that they will speed up and go. So like the Ravager, I think, is the biggest mob or heaviest mob that we can put in a minecart. And I believe when we go up. See, we are still gaining quite a bit of speed. I don't know if I have to set to it. It will shoot us straight back down though. Oh. But what do we hit on the Y coordinate? -60 to 500, almost 600. Okay, so not quite as high as with the camel, but still pretty daggum high. Well, I hope you guys enjoyed this fun little video today.
Wow. Okay. Hello, Leon Plays. I was a little bit distracted there, guys, but we gotta stay on task here. Primary structure versus secondary structure of protein. Here we go. Protein structure. Organic chemistry tutor. Ooh, let's take a look.
In this video, we're going to talk about the structure of proteins. And what basically is a protein? A protein is a polymer consistent of many amino acids. So each amino acid represented by the circle is a monomer that forms the protein. Whenever you have many amino acids, it's also called a polypeptide.
Yes.
And the bond that
Perfect.
connects each individual amino acid residue is a peptide.
Peptide bond. Peptide bond. He's gonna say peptide bond.
Bond.
Yes.
So let's talk about the
okay.
individual
Oh,
structure
sorry, guys. I'm
of an amino
actually
acid.
learning something. Oh my god.
So an amino acid has
I'm actually
a
learning something
chirocarbon
here.
with a hydrogen attached to it, and it has an amine group attached to it as well.
Yes. Cool. Got it.
And then it has an R group,
Essential carbon, R group in the carboxyl group.
and next to it, a carboxyl group.
There we go.
Acid. The right side that has the carboxyl group is the C terminal of the amino acid. Now, these two amino acids can react with
C terminus. C terminus is going to be at the right side. or no, the carboxyl end is going to be the C-terminus.
With each other in a condensation reaction where we're going to lose water and we're going to connect.
That is, what's it called? Dehydration synthesis.
The two amino acids into one molecule. So once water is reaction,
Takes that out, and
Fit
then
it
we're
in
going to be
here.
left with a nitrogen, right? We're going to be left with nitrogen.
we lost two.
Yes, with this.
We have one hydrogen left over. We lost two once we lost water. So this is a dipeptide because we have two amino acids combined. And so this amide bond that we see here, that is the peptide bond. We formed a bond between the carbon and the nitrogen atom. So whenever you combine two amino acids to form a peptide bond, you're forming a covalent bond, which is hard to break. Now let's talk about the different levels of protein structure. There's 4 levels that you need to know: the primary structure, the secondary structure, the tertiary structure, and finally the quaternary structure. Now the primary structure is simply based on the sequence of the amino acids found in the protein. The sequence determines the shape of the protein. If you replace just one amino acid with another, it will completely change the shape of the protein. So the shape and its function is primarily determined by the sequence of amino acids. The secondary structure describes the localized shape of a protein, and there's 2 of them that you need to be familiar with: the alpha helix and the beta pleated sheet. In the alpha helix, you can see that it's stabilized by hydrogen bonds. The NH group of one amino acid interacts with the carbonyl group of another amino acid, and each turn contains about 3.6 amino acid residues. Now here is a visual illustration of the beta pleated sheet. Just like the alpha helix, it too is stabilized by hydrogen bonds between the carbonyl group of one amino acid and the NH group of another amino acid. Next up, we have the tertiary structure, which represents the 3-dimensional complete folding pattern of the protein. And so here's a visual illustration of it. So we can see
Explain which one? Just elaborate a little bit.
some areas it's just a straight chain. Here we have an alpha helix, and here we have a beta pleated sheet. Now, the tertiary structure is one individual subunit. When you combine multiple subunits, you create a quaternary structure. So hemoglobin is an example of that. It has
I get it now. The video so far, what you want? Wait to the end. Oh,
4 individual
give me
subunits,
1 second.
2 alpha subunits and 2 beta subunits. So hemoglobin is a protein with a quaternary structure. And so that's basically it for this video. Hopefully it will completely change the shape of the protein. So the shape and its function is primarily determined by the sequence of amino acids. The secondary structure describes the localized shape of a protein, and there's 2 of them that you need to be familiar with: the alpha helix and the beta pleated sheet. In the alpha helix, you can see that it's stabilized by hydrogen bonds. The NH group of one amino acid interacts with the carbonyl group of another amino acid, and each turn contains about 3.6 amino acid residues. Now here is a visual illustration of the beta pleated sheet. Just like the alpha helix, it too is stabilized by hydrogen bonds between the carbonyl group of one amino acid and the NH group of another amino acid. Next up, we have the tertiary structure, which represents the 3-dimensional complete folding pattern of the protein. And so here's a visual illustration of it. So we can see some areas it's just a straight chain. Here we have an alpha helix, and here we have a beta pleated sheet. Now, the tertiary structure, the alpha...
Okay, I just need to understand the secondary structure and the primary and the secondary structure. I get what the tertiary structure means. In the quaternary structure, it's just a bunch of primary and secondary structures just mushed up together, pretty much, which would create a quaternary. And maybe a little bit in the tertiary, but let's see if we can find a video that explains this a little bit more rapidly. Primary and secondary structure. Amoeba Sisters, that's a classic.
Let's just say I invest a lot in anti-wrinkle launder spray. Amazing invention. My issue with folding extends to paper, too. I know foldables in the classroom can be a powerful way to organize concepts, But for me, it was the actual folding part that I tended to get stuck on. You may think of folding as a convenience, of a way to take something and make it more organized or condensed so it doesn't have to take up so much space. This is true, but in biology, folding can actually have a lot to do with function. We've mentioned how amazing proteins are. They can play so many roles. They can make up channels, be part of a structure, serve as enzymes for important biological processes, be involved with protecting the body, just to name a few. We've also mentioned that you are making proteins all the time in a process known as protein synthesis. But the conclusion of producing a long chain of amino acids doesn't necessarily equal a functional protein. There are modifications to a protein that often need to happen in order for it to be functional. By modifications, we can mean many things. It might be adding certain chemical groups, such as phosphorylation, something to definitely explore. But another important event to make a functional protein is, believe it or not, folding. But before we get into protein folding, let's talk about shape and why shape is so important. Shape and function in biology frequently go hand in hand. In our cell signaling video, we mentioned how protein receptors and the signal molecules that bind them can fit together so perfectly to start some type of cellular response. Or in our enzyme video, we talk about how enzymes, which are frequently proteins, have a very specific shape for the substrates that they build up or break down. When we talk about the way proteins are folded, we need to understand the different levels of protein structure, because there are different ways of folding that can happen in the different structural levels. The first level of protein structure is primary structure. This is the sequence of amino acids that make up a protein. Amino acids are the monomer, which means the building block, of a protein.
I see. Okay. This is a primary structure, right? This is a primary structure polypeptide. This is a polypeptide. I'm pretty sure this is a polypeptide right here. Okay? Gotta make sure we learn that vocabulary too. Why is my voice so nonchalant? What?
They are held together by peptide bonds. In protein synthesis, amino acids are added to...
I don't know if my voice is that nonchalant. I'm gonna be honest. Might be just how it is. Anyways, continuing on with the video. I think I have to study for the SAT soon, so let's finish up this video and then we're gonna get to SAT.
Form a polypeptide chain, and proteins are made of one or more of these polypeptide chains.
Yes, October 5th is my SAT.
Genes, which are made of DNA, determine the order and number of these amino acids. That sequence is critical to the protein's structure and function. In our mutations video, we talk about how one amino acid can be changed in sickle cell disease. Even a single change of an amino acid has the potential to affect a... So even though we have them drawn here like a chain of circles, realize that each of these circles we're drawing is structure. Folding is really going to start to happen. In secondary structure, the sequence of amino acids that we mentioned in primary structure
Sorry, my microphone is on. Off. This is a alpha helix. This is beta plated. I have to remember that.
can fold in different ways. The most common ways are the alpha helix and the beta pleated sheet. And which one of those foldings the protein does depends on the amino acid arrangement it has. Both of these shapes are due largely in part to hydrogen bonds. Those hydrogen bonds can occur at specific areas of the protein's amino acids. Specifically, these are hydrogen bonds involving the backbone of the amino acid structure. On to tertiary structure. This is looking at more folding that occurs in the 3D shape of a functional protein. And a lot of this is due to something we haven't mentioned much, the R groups, also called side chains. See, the amino group and the carboxyl group are generally standard parts of an amino acid, although the R group found in amino acids can vary among different amino acids. That means the R group can define the amino acid and can make amino acids behave a certain way. For example, some R groups are hydrophilic. They like water. Some R groups are hydrophobic. They don't. And remember that proteins contain many amino acids, which can contain different R groups, and so different areas of the protein can therefore be impacted based on those R groups. When protein folding is going on, amino acids with hydrophilic R groups may hang out on the outside, while hydrophobic R groups, where are they? They might hang out at the inside part of the protein. The 3D shape is due to other interactions besides hydrophobic interactions. Ionic bonds, van der Waals interactions, disulfide bonds, and hydrogen bonds, all involving the R groups, can influence the fold. Occurring in tertiary structure, something to explore. Now, when we've been talking about a protein, we've been talking about a polypeptide chain that has been folded into a functional protein. But proteins can be made of one or more polypeptide chains. And in quaternary structure, you are looking at a protein consisting of more than one polypeptide chain. Each of these polypeptide chains can be a subunit, and interactions between them, such as hydrogen bonds or disulfide bonds, can keep them together. Going back to folding, I know what you might be thinking. Who is doing this folding anyway? Are the proteins just folding themselves? Well, the interactions mentioned, like hydrogen bonds and R group interactions, are occurring depending on the protein's own amino acids. One reason why amino acid sequences are very important for protein function. But folding is far more complex than that, and there can be intermediate steps involved when a protein is folding. In fact, there's a phrase you can search called the protein folding problem to learn more about the questions scientists continue to explore regarding protein folding. Research has shown that proteins often have help in the folding process. Chaperonins, for example, are proteins that can help with the folding process. They have almost a barrel shape. Proteins go into them, and the chaperonin tends to have an environment that is ideal for the protein's folding, and this can help the protein be folded correctly so it's functional. Just wish I had something like that for my towels. All of these interactions we mentioned in primary, secondary, tertiary, and quaternary structure are paramount for a mature protein to have its correct shape so it can carry out its function. And that's very relevant. There are many diseases that are related to protein misfoldings. Check out some of our further reading suggestions in the description about that. One last thing we haven't mentioned. Each protein has an ideal environment for functioning, which might include a certain temperature or pH range. If the protein is exposed to something outside of its ideal temperature or pH range, exposed to...
Okay, I get it now. If you guys don't get it, I'll explain it real quick. So, primary structure. That is just our classic polypeptide chain, right? Polypeptide chain, peptide bonds, all that stuff. That will make up our... Hello, Jacob Linder. Yeah, that is going to be our primary structure. Our secondary structure, at least the ones we need to know, is the alpha helix and the beta. Yeah, the alpha helix structure and the beta plated. Made up with hydrogen bonds. I think it's hydrogen bonds. Double check.
And the beta pleated sheet, hydrogen bond.
Hydrogen bonds. Okay, nice. This is made up with hydrogen bonds. That's the same with beta plated sheets, but just this way now. This is it's like a paper map being folded. Thank you. Very nice to hear that people are genuinely interested. Very heartwarming. I have never gotten this amount of people on my stream. Oh, no. You don't really... Pre-AP Bio. That's probably like an extra class that's provided in your school, but my school doesn't have that. We just go from bio to AP Bio. I think that's probably just the regular biology class, but you have to learn chemistry too if you want to understand this stuff, which I'm assuming pre-AP Bio, maybe it's like a freshman class, I'm assuming. At least in my school, it's a freshman class. Oh, excuse me. Okay, anyways, I hate this. We are now moving on to... Now, gonna move on to... SAT. The moment you guys all been waiting for. Something I really need to start doing. Alright, let's make this fast. Make that smaller. Okay, make that a little bit bigger. Okay, going to enter my password and stuff. Please hurry up. I have to get to bed very soon. Very, very soon. Wait, on Thursday? 2 to 3 hours? Oh, that's good. Okay, I won't be on that. Score details. Alright, we were still continuing off from yesterday with the math section. Math. Cool. We're on the second part already, so it's, I can't remember which one. Was it that one? I reviewed this one already. Yeah, I think this is the last one I left it off at. Okay, so we're on this one now. Okay, so a proposal for a new library was sorry. Let's close this out, please. Okay, anyways, sorry. A proposal for a new library was included on an election ballot. A radio show stated that 3 times as many people voted in favor of the proposal as people who voted against it. So a radio show stated that 3 times as many people voted in favor of the proposal as people who voted against it. Okay? A social media post reported that 15,000 more people voted in favor of the proposal than voted against it. I chose what? I chose D, okay. I just multiplied 3 of 15,000. This was really confusing. This was really one of the really weird, oddly worded questions. Might gonna need help with this one, because I actually spent a long time on this one. Okay, based on the data, how many people voted against the proposal? So, stated that 3 times as many people voted in favor. 15,000 people voted in favor. 3 times as many people voted in favor. I was thinking like 15,000 divided by 3. Let's see what that is. 5,000. We have none. That is 5,000. Based on the data, how many people voted against the proposal? So many voted. 15,000 more people voted in favor of the proposal than voted against it. So 15,000 more. Okay, so 15,000. Okay, let's actually write this out here. Oh wow, that is fat. Let's use this one here. Let's actually use a different color here. I don't like this color. Not very appealing for my eyes. How do I change? Oh, here it is. Okay, let's choose black. Just pure black. Black, please. Black. Here we go. Okay, so let's do this. Okay, so 15,000 people. Hello, Got Milk. Okay, 15,000 people. More people. So more people. I need to shift this over. Shift it over a little bit. I need to please. How do I select here? How do I move? Oh, there. Okay. Okay, so 15,000 people. Subtract with X. It's gonna be our total. Right? Voted in favor. No. X is gonna be our number of voters who voted against. Plus 15,000 is gonna equal Y. Alright, and then a radio show stated that 3 times as many people voted in favor of the proposal as people who voted against it. Okay, voted in favor, so 3 times as many people. I'm thinking of I think this is a systems of equations question. I'm not too sure. Social media post reported that 15,000 more people voted in favor of the proposal than voted against it. So this is true. So this would be the total number of voters. Okay, so that is going to be total number of voters. So Y divided by 3 is going to equal number of voters who voted against. Why are 3. Okay, wait. Social media posts reported that 15,000 more people voted in favor of the proposal than against it. Based on this data, how many people voted against? 3 times as many people voted in favor of the proposal as people who voted against it. Okay, yeah, I don't get it, so let's actually see the answer here. Choice A is correct. It's given that a radio show stated 3 times as many people voted in favor of the proposal as people who voted against it. Next, we have the number of people who voted against the proposal. Oh yeah, so this is a systems of equation question. Okay, yeah, let's actually redo this real quick. I'm gonna redo this. Let's see if I can solve it on my own. Considering this is a systems of equations. 3 times as many. So x, let's have x be our number total of people who voted. 4, okay? So x plus, no, times 3, no, 3x. It's gonna equal, no, 3, no, okay, so y divided by 3. I think that's it. And if any of you guys understand this, I don't. I don't really. I just know this is a systems of equations question, which I didn't realize when I started the question, when I actually did this question. Okay, so reported that 15,000 more people voted in favor. Yeah, I need to see it again. I don't know. Who voted against the proposal? False that 3x the number of people who voted in favor. Who voted in favor of the proposal? And 3x minus x, or 2x, is how many people... 3x minus x is how many people voted in favor of the proposal and voted against it. Wait. 3x minus x is how many more people voted in favor than voted against. 3 times as many voted in favor as people who voted against it. So if we let x number represent the number of people who voted against. Oh, x is the number of people who voted against. Okay, so x is equal against. Okay, now, let's see, well, we have that hint. Let's see if we can solve it on our own. So, reported that 15,000. But why minus x? Hold on. Minus x. It's also given that a social media post reported that 15,000 more people voted in favor of the proposal than voted against it. Thus, 2x is equal to 15,000. Let's see, this is how many people voted in favor. How many people voted against the proposal? Okay, so let's take a look again. Why is it 3x minus x? So 3 times as many people voted in favor as people who voted against. So that would be 3x. Oh, that would just be 3x, right? 3x is equal to 15,000. Radio show stated that 3 times as many people voted in favor of the proposal as people who voted against. I think what it's saying is that when we multiply it by 3 times, it's actually like including. I need an explanation. I needed a very thorough explanation here. Let's copy this.
So this question is a pretty long question. Is based on these data, how many people voted against the proposal? So I'm going to actually use a strategy here called plug-in answers. Voted for, so I'm going to go 7,500 plus 15,000 times the number of the against votes. Also,
Are you struggling in your calculus class?
I'm not.
Do you have a hard time understanding your instructor? Do you
Number 15, a proposed more voted in favor. So that original number X, 15,000 was added to it, and it equaled those in favor of it, which we also said was 3X. Now the question they're asking us is, based on these data, how many people voted against it? So we're solving for X, and with our little
This would result in the answer of 7,500. But where exactly do we get this equation? Okay, so X is going to be our total number of against, plus 15,000. Oh, more people. Okay, so X plus 15,000. So this would be our more people statement here. So the social media post reported that 15,000 more people, right, voted in favor. A radio show stated that 3 times as many people voted in favor of the proposal than those people who voted against. It's people who voted against. So our total number of people. Oh no. Oh yes. Okay, I get it now. Hello, Jay Stephens. I get it now. Okay. So here we have 2 equations here. This equation represents the number of these both sides equal the number of against, right? Number of against. This was very confusing. So this X plus 15,000 represents the number of against. 3x, or 3 times X, represents the number of against. Or actually, X is equal to the number of against. Right? And both of these equations are perfectly equal to each other. So that's really where we get 2x is equal to 15,000. And then we will get X is equal to 7,500. I see now. Okay, so I just needed to structure it like this a little bit more instead of a system of equations. Okay, got it. Anyways, let's see, this one here. Okay, let's hide this answer real quick. Caleb used juices to make popsicles. Okay, let's write our function first. See, f of x equals negative 5x plus 30. Approximates the volume in fluid ounces of juice Caleb had, in fluid ounces of juice Caleb had remaining after making X popsicles. So X is equal to popsicles. Right, pop. Right. Which statement is the best interpretation of the y-intercept of the graph y is equal to f of x in the xy plane of this context? The best interpretation of the y-intercept of the graph. The y-intercept. So our y-intercept is going to be the beginning point, right? Wherever it is, it's going to be at 0. Y-intercept, always at 0. So it's going to be at the beginning of something. So he add, I think I said had, or did I say C or D? I chose A. Okay, yeah, that was pretty stupid. Now that I actually reread this question, this answer should actually be, I don't know about that, but I mean the algorithm is kind of weird, you know? Like sometimes I get like pretty, you know, I'm not, honestly, I don't think I'm that funny to be famous. I think you gotta be like pretty, you have to be interesting to be famous. I don't think I'm that interesting. I'm just a regular guy who just studies, you know, does game development and stuff. Anyways, okay, so had approximately 30 ounces of juice when he began. Okay, yeah, well, this 30 right here obviously represents our y-intercept, and as he makes more popsicles, it's gonna get lower and lower and lower. So it should actually be C. And yeah, I don't know why I chose that. I probably didn't read through the question right. Omitted. Okay, yeah, I didn't actually finish. So let's just review our incorrect answers, and we can go over the. Oh yeah, only go over through our incorrect answers here. Okay, so dataset A frequency, dataset B frequency. Dataset A and dataset B each consist of 27 values. Okay. Table shows the frequencies of the values for each dataset. Which of the following statements best compares the means of the two datasets? The means of the two datasets. Which of the following statements compare? Okay, dataset A, dataset B consists of 27 values. 27 values. I'm assuming 2 plus 4 plus 5 plus 7 plus 9 is going to be... I select C. Oh, there's our... Okay. Select C is equals to the mean. I can see why, because all I did was just add these points up, and just because they both equal 27 values, right? And then they both equal 27, so obviously the mean is going to be the same, right? So let's see. Which of the following statements best compares the means of the two data sets? Okay. Consists of 27 values. The mean? Oh. Okay. I think I might have to subtract 2 from 30 and 9 from 30, and then do that for the rest of these values, and then compare the means together. Oh my gosh, there's a bug here. Anyways, okay, so let's actually pop up Desmos here. I'm gonna actually leave this back in here. Hello, Ray. I am currently trying to solve this on my own before asking for help, of course. Okay, so 30 minus 2. Black screen. Oh crap, I was literally just talking the entire time. What the freak? Why didn't anyone say anything? What? What the hell? Wait, did it just turn black? Or was it black the entire time? How long was it black? What the heck? Anyways, okay, so we have 4, 5, no, 34, 4. Oh my gosh. What the heck? Why is OBS actually tweaking on me?
That's why you say you're busy.
38. Okay.
You say you're busy studying.
I am. I am.
Turn on the light, man.
What the heck are you talking about?
Yeah, that's why you're busy. I
Yeah.
say you're busy.
Yeah,
Yeah.
that's what I've been doing the
Of
entire
course.
time.
What did I say?
I mean, this sounds like a sarcastic tone, but
Okay. What do we then?
42 minus 7. Okay, I'm thinking I have to grab the mean of all these values. At least that's what I think. Let's get this out of here. Oh shoot.
What was your first part?
Alright guys, hold on. Black screen. I'm going to remember to unblack the black screen. Give me 1 second. Show me the question. Yeah, give me 1 second. Give me 1 second.
Yeah.
Hold on.
You got the job.
That's actually outrageous. 17 minutes. 17 minutes. I'm actually like, bro. Crap. Where's my practice scores?
Yeah.
Where is my testing scores? Start practicing.
Yeah.
Okay, Daddy. What was your favorite part?
Yeah, I know. I'm trying to find my practice stats. I lost it. hold on, hold on. I am trying to find. Alright, might as well just open Blue Book. I'm sorry, everyone.
Oh, okay. Yeah.
Bowling practice. No, I want to see my score. Pass. Here, practice too. Here are my responses. Here we go. Alright, I'm gonna close this. I'm gonna unblink my screen. I'm blinking my screen now. There we go. Okay. Alright, score details. Back to where we were. I should have just opened history actually. You see how stupid I am? Okay. 5, 4, 4. Here. Incorrect. Here's our data set. You're gonna pause the thing if you want. Take a screenshot. Do whatever. I'm trying to see if I can do it myself. 42 minus 7, 46 and 9. 46 and 9. So 28 plus 30 plus 33 plus 35 plus 37 divided by 5. 133.4. Nice. So this would be our first average. 30.
Yeah.
30 minus 9. 34 plus 7. No, minus 7. Sorry. 38, 5. 38 minus 5. Excuse me. 42, 4, 46, 2. 42, 4, 46 minus 2. Oh, 42, 46. 38, there we go.
No,
Okay,
too
it's
far.
21 plus 27
Wrong.
plus 23 plus 38 plus 44. It's 152. 152 divided by 5. 38.4.
Yep, yep. Go down one go.
Oh, wait, no, hold on. There, 163. 163 over 5. 32.6.
TH.
So,
Không nhớ.
data set A is going to have a larger, a greater mean. Should be A, right? Yes. Yes. Oh, well, they multiplied it. The frequency, well, I mean, subtracting it, I guess, would...
PAGE.
27, what? Well, what? You got it right, but not the right way. That's not good. Not a great sign. So, let's see. When a data set is represented in a frequency table, the sum of the values of the data set is the sum of the products of each value in its frequency. Okay. Oh, they literally meant, like, literally frequency, like how many times it appears in the data set. So 30 appears 2 times, 30 appears 9 times. Oh my gosh, I'm actually so dumb. I didn't actually think they would, like, I thought they meant like frequency as in like a unit or something, like 2 megahertz or something like that. Like they literally meant like how many times it appears in the data set. Right? So they do 32. And this would obviously equal to 27.
And 1.
Number, etc. Now let me try that.
Yes, because it is easy to read.
1, 2, 3, 4, 8.
It says yes.
Her?
It is good. Yes, because
Oh, that's so cool. That's actually cool. Great to know. Yeah, got it. Okay, well, yeah, I understand now. So this is the total number of data points, right? gonna have to do this, the frequency, this with the frequency, this with the frequency. Okay, I get it now. I understand. We don't have much time left, so I'm just gonna go over these other questions. Let's hide the correct answer.
I just spelled easy.
Okay, let's go over the answers that I didn't finish. Okay, so we have x plus 1. Spread out our equation first. 56 is equal to 4x. X minus 7. Okay, what is the sum of the solutions to the given equation? Excuse me. Can I have my answer choices, please? Well, that's unfortunate. I can't see my answer choices, so I can't really say anything right here. Well, let's try and simplify it. Desmos. Do this. I can simplify in Desmos too. I think it'll only show the graph though. It's the only thing that. Okay, so let's actually simplify it. Minus 56 is equal to 4x squared minus 28x. It's always my first instinct. So negative. No, let's actually do it on this. No, let's keep it inside. Negative 3x squared plus 29x minus 5 6 equals 0. X squared. You can do y and then. Yeah, I know, but like that's like. Or yeah. Oh well. No, I think that would be a solution to the system of equations. Add the x values together. Huh.
That's 2040.
Let's see.
You want to spell it again?
Wait. Nope. Okay. Nope. Okay. Okay. Yeah, spell it again.
There you go.
Now, can you do Paris signature first? Paris signature. Paris signature.
Yeah.
But there's no intercept.
Check if everything is right.
There's like
and
zero
check if I didn't miss
intercepts.
anything.
What is that? CIU.
That's why.
It would be 28x
Oh.
Proof.
squared. Oh yeah, wait. What? What the heck? I'm actually stupid. You're right. Oh, and then we add these points together. So 7 and 0. So it'd be 7.
Let's practice spelling.
But how would I do it in paper? If I didn't have Desmos, how would I do this on paper? This is the question. That's what I'm trying to do here. Right?
Yeah.
This is the whole paper that we can practice.
Okay. Put your name down.
If we run out paper, we take a new one. Get out of it.
Okay. You ready?
Yeah.
Rash.
And then scroll down.
So you put the book in the backpack tomorrow?
Oh.
You broke this?
No, it was actually like that.
Okay.
Oh, it's just that 0.0. Oh, that's okay. That makes sense.
Is this a good book to begin?
Well, it looks like it's a big fat decimal.
Okay. Is it?
Okay. Rash.
What?
But like, how would I do this in paper is the question.
The cow eat what?
Without using Desmos, of course.
I keep hearing you say it with the R.
So I do have this equation here.
I keep hearing you say it with the R.
Okay.
G.
I don't think I can do much.
A.
No.
G. R. A.
No.
S.
If you could help me out here. Try grouping.
Grass.
It's grouping.
Grass. There's a G, right?
How to say that?
Grass.
Grass. Good. Grass.
But you don't know a lot of English because you're doing that. Okay, anyways, let's continue.
Yes, I'm looking to factor it.
Strip.
Trip.
S mutable trip. Yeah.
I see.
Table crack.
Oh,
Okay, so we could do.
Oh, yeah.
now look. Crack.
Wait.
And multiple cracks or just one crack?
What crack?
Okay.
Steel.
What?
Do you still?
Yeah, I know. 29 isn't a right number, but like in Desmos, there's obviously
Ew,
two
what is
intersecting
it?
points.
That's a table.
Oh, this is scary. Tưởng là mũi bay.
Steel, right?
Đúng rồi, good. Yep.
So is there any way to do it on paper? Is just
Yes.
there's only Desmos?
Okay, west.
I don't think that matters.
West.
Yeah.
Yeah, I don't. Maybe not actually. Try
That's
to
it,
add to
west.
equal 29. Yeah.
That's
Well,
west.
wait. Hold on.
Only
I have to remember
Is
my
only
equation
E.
again. Hold on.
Okay,
It
close
was
enough.
definitely.
you wrote that one last week too.
Really? Oh yeah, I did.
Negative
Okay.
3x squared
Mass.
Math.
Mass.
minus 29x minus 36.
Okay, next.
This is definitely better though, because yeah, well, that's true.
Next.
But like, what if I didn't want to use it though? Because I'm kind of like stubborn like that. I want to do it on paper. Unless there's like no possible way, I would probably give up. But like, I think there's a way to do it. Yeah, I know. I probably will use it.
That's clever.
Yeah, I know. But like, if I didn't have Desmos with me, right? How would I do it? Is the question.
Yeah. See.
I understand this might be a waste of time to you, but I truly want to be able to do this.
Remember this. 1 L only.
Yep.
I'll try dividing it by 3. Looking at 3 here, negative 3
It
at
doesn't
least,
look like it.
plus.
Are you sure this is correct?
Man, why are you so noisy?
Yep. Okay. Next.
9.66667.
Plans. Do you have plans?
9.66667.
Correct? Or no?
9.66667
Mutable
is
plans.
equivalent to what?
That's not the word.
This is correct, right?
Yeah. What else?
Plans. That's
NS. NS.
N. Plans.
Wait,
Milk.
wait, no, this is plus. Sorry, this is plus. You're right. You're right.
Milk.
You're right. You are right. I'm actually tweaking.
Okay, next.
Anyways,
Belt.
What?
Belt.
Belt. Like a belt goes around.
Yep.
so what if I plug this equation in? Negative 3x squared
A.
plus 29x
It makes
minus
sense.
56.
Yeah.
Oh, interesting. Oh, it's actually interesting. You can see that. If I plug that in, the 2 points would be these two added together.
LT.
Notice that.
Okay, next.
Far. Yeah.
Okay, I'm doing a little worse.
You know, I literally could have what I could have done is
Top.
I mean that's true, but like
Top.
it's just how to do it. It explains it. Yeah, I should do that right now. I mean, I was gonna do that anyways, but like considering I have someone helping me in the chat right now, so.
TUB. Đúng rồi.
Is it next spot?
Hmm?
Bye next.
Yeah.
How do I copy?
Next.
E.
Next.
C.
Okay. Okay, so
look.
x
I did a
x
little bad.
plus 1 minus. Oh, there it is. Wow.
I did a little
Wow.
bad. I got trips. But I got trips correct. Press yes, still yes. Was wrong.
Grammarly is a must-have for students. Grammarly has this generative AI tool that can.
She sounds like she
So this
wants to
question
die.
looks like it's going to be a simplified question. C, all over 2A.
Don't hit your head.
Oh, oh, he uses. Oh, I literally forgot about that. What the heck? I actually forgot about that. Oh my gosh. Oh my gosh. I bet you guys forgot about it too. This is the this classic equation. Negative b plus minus b ac. I think I have to remember this equation.
B squared minus 4AC.
B squared minus 4ac. There we go. B squared minus 4ac. Divided by 2a. Right? 2 or 2b? 2a or 2b? Can't remember. 2a. Okay. Well, let's see if we can do it like this. This is probably like this is what I'm talking about, guys. I like want to do it like the mathematical, like quadratic form. I know. It's like I don't want to. It's like I want to remember it, but it's not that easy to remember. Right. So okay, we're gonna plug in our values here. Okay. Let's put this on the side. So negative 29 plus minus square root of 29 squared. Make sure that this is absolutely pinpoint accurate for negative 3 times negative 56. And square root October 5th is when I'm taking it. I have to absolutely study the hell out of this. So negative 3. There we go. And then just solve it. I'm actually gonna do this on paper. This is like. Yes, thank you. I cannot do this. I have to do it on paper, guys. This is actually a waste of time. Those are all like the unused ones, Mom. Like the not very used. Yeah. The school makes me buy these big 3-subject notebooks, and we just never use them. Huh? I don't know. That's the box of supplies. Okay. Anyways. Huh? Sign up for a different date? Hell no, bro. I'm confident. I'm literally confident that I can. Like, I'm confident that I can study hard enough that. Come on. Anyways, let's continue on. This is just, it's taking me a lot slower because I'm like trying to explain it and stream it as well at the same time. Because I probably would do this a lot faster if I wasn't streaming. What grade is the SAT for? The SAT is usually after sophomore year. You gotta like know everything of Algebra 1, Algebra 2, Geometry, and Trigonometry is what you should have already known. Okay, let me just keep solving for this. Give me 1 second. If you guys are wondering and are solving this with me, it's gonna be there's something that could help. 4-1 minus 12. No wait, crap. I don't have a freaking eraser. I threw my eraser at that bug. Okay, so let's do 3 times 56. 56, 168. 168, 4 times 168. Okay, 4 times 168 is 672. So negative 29 plus minus square root of 841 minus 672 over 2a, which is 2a, which is going to be negative 3, right? AC, yeah, so negative 3. So it'll be negative 6, over negative 6. Think. 11 and 12. AC, A, negative 3. Okay, cool. Alright, preferably 12th when you're less busy with work. I mean, well, yeah, I guess you could say that, but I'm just taking it because I want to do everything now. I want to chill out my senior year. I want to give myself time to rest. Because I hate, like, I know I have to put in the work at some point, right? And I'm willing to put it in. I feel like 12th year doing college apps, so it's stressful. No, do 11th. Come on, 11th grade. If you're doing college apps and you're saying it's stressful, then obviously that proves my point that you should take it earlier. Anyways, I need to finish this equation. Okay, so squirt of 841 minus 672. 13. Wow, that's actually a great number. Luckily, it's a very precise number. Plus minus 13 over negative 6. All right, this is what I got. If you guys are, after doing all this, I got negative 29 plus minus 13. Plus minus 13 over negative 6. That is what I got. And I'm trying to solve for the rest. So I'm going to do addition first. Negative 42 over 6. Negative 42 over 6. It's a negative 7. That's weird. I think I should have. What the heck? Negative 7? That's not right. Guys, something is not right here. Oh, wait, no, wait. I forgot to write. Okay, yeah, so it is 7. Nope. Yeah, that's right. Okay, yes. Okay, so I have 7, which is this point right here. I literally could have plugged this into Desmos. And I could have actually, like, solved this a lot. Now, if you take AP Lang, it's not necessarily that hard. You can easily write like a 2-page essay and just... Yeah, surprisingly, I think that's actually not one of my most difficult things to do. Anyways, let's keep going. I have 7 for my first value. Takes like 2 hours max. Yeah, I agree. If you really want a good essay, maybe like take 1 day, like 1 full day on it, revisions, all that. No, not necessarily 2 hours max. Potato. Well, I mean, I can't really say anything because I haven't actually written a college essay before. But I don't think so, at least. Anyways, I gotta do my other part. It's gonna be plus 13 divided by negative 16 divided by 6. Right? Negative 16 divided by 6. No, negative 6. It's 2.666667. And we should have 9.666667. There we go. Alright, nice. Nice. Wow, this stupid... Look at this, a quadratic equation. Look at this, it's so annoying. Okay, anyways, the function f of x is going to equal... Oh, no, I don't want to see the answer. Get away. F of x is equal to 1/9 over x. Okay, let's write it down. I'm going to do it over here for you guys, actually. Alright. Draw. Okay, this is a very important. I want to make sure I remember it right now. Discriminant, cool. I remember the discriminant thing. B squared minus 4ac. B squared minus 4ac. There you go. And that would determine the amount of solutions. Right, and then for solving polynomials. So we're going to get a lot of those. So it's going to be negative b plus minus squirt b squared minus 4ac over 2. Or actually just underline it. What the heck? for AC. For AC over 2. Just use Desmos. Yeah, I know. I will. I'm gonna have to practice doing that, but I want to make sure that I know this, like just in case something goes wrong, which is probably not gonna happen. I know. It does save time. Like you could just look at the points and just add them up together. Easy. Saves a bunch of time. I understand, but just in case I get stuck, you know, I want to make sure I understand. Solving polynomials, when adding, can add the two x-intercepts, right? X-intercepts? Yeah, the two x-intercepts of the added together / simplified equation. Only if it results in a polynomial. But it's best to just put 2 halves of the equation into Desmos and add the points of intersection. Hello, Ismail. Okay, right. So that's gonna be our equation. B squared minus 4ac. I think that's right? That's our equation. Our equation. Need to erase all this. Let's see. Yes, okay. I have memorized it. Okay, so negative b plus minus square root of b squared minus 4ac divided by 2a. Yes. Okay. Function f of x. It's probably gonna be the last one before I head to bed, guys. I'm getting a little tired. So get ready for school tomorrow. You call a blind man with no legs? What? What do you call a blind man with no legs? Plus 3 gives a metal ball's height. Okay. What? I don't get it. Anyways, so f of x is going to equal... Okay, ball's height is equal to ball's height. Ball's height above ground. Okay, we're given an inequality here. Which of the following is the best interpretation of the vertex of the graph? Y is equal to f of x on the xy plane. Okay, so let's do it the Desmos way first, like you guys have mentioned. Let's get rid of all this. F of x is going to equal 1/9. 1/9. Not a very active stream, guys. This is just random people. It's actually the most amount of viewers I've had in a while. Very occasionally get that. Okay, so this is cubed. This is squared plus 3. Plus 3. Okay. 0 is less than or equal to x is less than or equal to 10. Oh, wow. I know, right? 3 viewers? Crazy. Just imagine there was like thousands of viewers. That'd actually be kind of crazy if I had thousands. But anyways, okay, so what is the best interpretation of the vertex of the graph? Y is equal to f of x in the xy plane. The metal ball's minimum height was 3 inches above the ground. The best interpretation. What's the best interpretation? Okay, so starting from 0, it was 8.44. When it started moving, 1400 plus. 1400 plus is my goal. In 1 week. In 1 week. And then mine is next week on Monday. No, Saturday. Saturday, October 5th. So we have plenty of time. So I have to make sure I do all my homework, no late assignments, and absolutely grind the hell out of this SAT. Let's see. You guys believe I can get a 1400 plus on the SAT? With just 1 week? Oh, okay, let's see. Which is the best interpretation? Grand Khan Academy. Yeah, Khan Academy, I heard Khan Academy was a great source too. But reviewing my answers here is also a good way of learning, my opinion. Remember, I'm also learning every time I fix my mistakes. F of x in inches, x seconds after moving of the track. 0 is less than or equal to x, less than or equal to 10. Which of the following is the best interpretation of the vertex of the graph? Vertex. So 3. The metal ball's minimum height was 3 inches above the ground. How does that make any sense, guys? It goes down and then it goes up? I think this is A here. No, yeah, minimum height was, I think this is A. Save a few questions, mod 2 math. Yeah. A, right? Yes, the answer choice is A. Okay, let's see how they did it. The form where f of x is equal to a1 over 9 is equal to 7, k is equal to 3. Therefore, the graph is a parabola that opens upward with the vertex 7 over 3. Okay, well they just plugged it into a graph. So that's pretty much what I did too. So yeah, that's solved. Okay, let's see. This is question 14. 14. I don't think I'll be able to finish all this. Let's do one last one before I head out. Let's see if we can do this one on Desmos as well. The function f gives the temperature in degrees Fahrenheit that corresponds to a temperature of x kelvins. If the temperature increased by 2.10 kelvins, by how much did the temperature increase in degrees Fahrenheit? Oh, this is easy. You just plug 2.10. Yeah, you just plug 2.10. We just put this in a calculator. This is easy. Wow, if I had an easy question like this. Okay, so let's do 9 over 5 times 2.1. I think it's 2.1. Yes, 2.1 minus 273.15. Minus 273.15. Plus 12, 32, is negative 455. Did I do that right? Increased by 455. That is odd. 9 over 5, 2.1, 273.15 plus 32. That's odd. Huh. 450. What if I put 0? Nope. Wow. That was stupid. 273.15 plus 30. Are you guys seeing anything? Because I don't think anything's wrong here. Corresponds to a temperature of x kelvins. Temperature increased by 210 kelvins. How much did the temperature increase in degrees Fahrenheit? How much did the temperature increase? 455. So it's probably C, right? Answer is A. In degrees. F gives the temperature in degrees Fahrenheit. That's weird. That shouldn't be right. No, 2.10 kelvins is really low. Minus 273.15 equals 32. Are you guys seeing anything? Hello? So I am so confused. The answer is A, 3.78. What? Wait, okay, the function f gives the temperature in degrees Fahrenheit that corresponds to a temperature of x kelvins. Okay, if the temperature increased by 210 kelvins, by how much did the temperature increase in degrees? What? That would make sense if they put this, but like, hello? Are you guys seeing anything? Okay, why don't I plug this to a graph? Okay, so this is all the way down here. That's odd. Oh my gosh, what is wrong? Here's the question. This equation represents the degrees in Fahrenheit that corresponds to the temperature of x kelvins. So it's pretty much a conversion rate, right? This is conversion. This is converting, right? If the temperature increased by 2.10 kelvins, by how much did the temperature increase in degrees Fahrenheit? Right? Excuse me. It's saying the answer is 3.78. Wait, in degrees Fahrenheit, the corresponds temperature increased. Wait. Yeah, wait, why? It says it gives the temperature in degrees Fahrenheit. What? It follows that an increase. Yeah, we want to see how much it increases. And it literally says if a temperature increased by 2.10 kelvins. No. Wait. Let's see. Well, there we go. There's our difference. But like, I still don't understand. F of x by 9 of 5, 2.10, or 3.7, the temperature increased by 2.10 kelvins, temperature increased by 3.78 degrees Fahrenheit. Degrees Fahrenheit. But they didn't say that it started at 0 degree Kelvin. Oh. Any number? Are you sure? F think so.
Now, can you turn off the light? It's still bright. Can you turn off the light? Is that okay?
Yeah, turn off the light, please.
Okay.
Far. Uh-huh. Yeah. Uh-huh. Oh. Sorry, Cole. I already stopped studying for AP Bio a while ago. We're pretty late. Started for the SAT now. Okay, so let's see. Judging by that, x equals 4, x equals 1, the difference will be the same. Okay, so if I did... Oh, I see. Okay, so if I did 2 plus f of 4.1 or minus, it would be 3.78. Okay. Okay, I get it. That's so confusing. See, these SAT questions are worded so weirdly. Temperature increased by 2.10 kelvins. Yeah, now that you said that you could take any value and that value increased by 2.10, that makes so much more sense. But I was thinking that... Wait, but if it literally increased by 2.10 kelvins, then why not? Wait, but if it increased... No, if it increased by 2.10, why can't we just put 2.10 into this equation and we would... Yeah, why? Why not? Why does it give us 455.89? Because if... Can you explain to me? I'm a little confused here. Why can't we just plug in... If it increased by 2.10, which is the difference, right? We just need to convert this degrees in kelvins to here. Oh my gosh, what the heck? Oh, that's so stupid. Oh my gosh, wow. My brain is expanding. Yeah, wow. Yeah, worded very weirdly. Okay, well, I think that's enough for today, guys. Thank you for watching. Oh my gosh. My butt's kind of sweaty right now. You need to get a break. I might, during the summer, I might play some more Roblox. I'm gonna stream that a lot, actually. Yeah, I'm definitely gonna be playing more, but during the school year, you're gonna have to study. It's straight study. No, gotta lock in, guys. You know, cannot start gaming now, but we will have our fun in the summer. We will have our fun, and I'm sure you guys will enjoy it too. Yes, I have to lock in. Gotta log in. Anyways, thank you guys for watching. Maybe tomorrow, maybe Blender tomorrow. Don't know. Maybe I'll get to Blender tomorrow. Maybe not, but definitely more SAT study. This is guaranteed tomorrow. But maybe not, because I have to go to the gym tomorrow. I don't know. Time's weird. Yeah, time's very weird. Well, we'll see. We'll see tomorrow. Yeah, hopefully we can start. Actually, no. Okay, I will stream tomorrow, but I don't know what I'll stream. Okay, so I will see you guys. All right. Thank you guys for watching. Adios.